Hello champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about translation of mRNA, and this is a very simple and a very interesting topic. So I hope you all are in loop and you have covered all the videos that we have done previously for this particular chapter, because this is a very high weightage chapter. Okay, now over here what did we cover the previous time? We spoke about the genetic code and I also gave you certain ways to remember it. I hope it was helpful for you. Okay, and the number of questions which came in the previous year was 6, but before that it was 10, and it had been going on alternatively like that. So we can expect much more number of questions this year as well. That is not this year, that is the next year, that is 2025. Yeah, and I believe my little chitkus solved this particular homework question. What would happen if in a gene encoding a polypeptide of 50 amino acids, so they have given an information that 50 amino acids are being formed with the help of this particular gene. So 50 amino acid means there will be 50 codons. There will be what, children? 50 codons will be there, and each codon will have 3 nucleotides. Okay, each codon basically will have 3 nucleotides. So 50 into 3, basically 150 nucleotides will be there. Okay, now they are saying on the 25th codon, that is UAU, is mutated to UAA. So UAU is now mutated to UAA. So obviously this particular codon might be coding for something else, one amino acid. This will be coding for some other amino acid. Right? Agree? Right? So what will happen? If this type of thing happens on the 25th codon, there is a mutation like this, then what can you expect out of it? I can expect that the rest 24 which were formed, since there is no mutation reported there, so there will be something that will be formed over here. This polypeptide will be all safe, and this polypeptide will be having 24 amino acids. Okay, it will be having 24 amino acids. Rest from this position onwards, I'm not sure what will happen. So this could be a bad protein. It might not be formed. Okay, that could happen, right? So a polypeptide of 24 amino acids will be formed, that is for sure, something that I'm sure of, right? Now, 2 polypeptides of 24 and 25 amino acid will be formed. No, I'm not sure of that. Okay, now next thing, and not sure of that, it's basically 50 amino acids, so obviously it does not make any sense. Now a polypeptide of 49 amino acids will be formed. No, children, I cannot be sure of that. A polypeptide of 25 amino acids will be formed. It's 24, because I'm sure of this particular thing, because it happened before the 25th codon. Everything was perfect according to the question. So this is the correct answer. This came in 2003. Okay, I hope you were able to solve this question. Now going ahead, today's thing. So first thing that we have to start off today is the structure of tRNA, and it's a very interesting topic. So tRNA, no children, tRNA says that I am the one that has the anticodon. So if you do not know the basics, let me explain you the basics a bit. So this is an mRNA. mRNA is formed with the help of a DNA. Okay, whatever information was there in the DNA, it has been transcribed to form an mRNA. This mRNA now moves out of the nucleus and goes and sees that, oh, there are ribosomes. Comes in between the ribosomes. Okay, so there will be ribosomes, comes in between the ribosomes, and on this ribosomes there are places where the tRNA comes, and the tRNA comes with something else that is shown over here. So over here, suppose this is the mRNA, and mRNA has UAC. This, I say, it's a codon. What is it, children? This is a codon. Now, something complementary to it comes and matches with it. And now this will be known as anticodon or anticodon. So basically the anticodon comes and matches with it, but what's the use of this? And what is this structure which is bringing this anticodon? This structure that you see over here is the one that comes and binds on the ribosome, on the mRNA, and does the translation process. This whole structure that you see, the twisted structure that you see, it is known as a tRNA. tRNA stands for, children, what? tRNA stands for transfer RNA. And what is it exactly transferring? It is exactly transferring something that is known as an amino acid. So this thing that you see over here is an amino acid. So you got to know one thing, that there is one area where the anticodon is there, and there is another area where the amino acid is being attached over there. Okay, and the structure is kind of twisted. It looks like this. Where is the amino acid attached? On the 3' end of the tRNA. This is also an RNA. So RNA does obviously have 5' and 3' end, right? So this is an RNA, and over here on the 3' end, what do we have, children? The amino acid. Okay, and over here we have the anticodon. coming in between. So now the thing is, you have to read and understand each point that is given in your NCERT, because from this question I have seen the direct questions have come from NCERT. No twisting, nothing. Okay. So the tRNA, which was then called sRNA. Why was it known as sRNA? Because you called it soluble RNA. Okay. The reason was it was soluble in some medium probably, which is why it was initially known as what? Soluble RNA. So the question can come, which is that RNA among them which was known as soluble RNA, mRNA, rRNA, this RNA, that RNA. It's basically tRNA, which was initially known as snRNA. Sorry, sRNA, not snRNA, sRNA. Please don't get confused. Now, tRNA has an anticodon loop. So this loop that you see over here, this loop is known as anticodon loop, that has bases complementary to the code, okay, and it also has an amino acid acceptor end. So this end that you see over there, it is known as amino acid acceptor end. So if I try to make this, I get an information like this. Okay, so over here, it looks somewhat like this. Okay, somewhat like this, where this is the 5' end and this is the 3' end, and over here I have the codons. Okay, so this loop, what I call it, anticodon loop. Fine. Now over here on the 3' end, what do I have, children? Over here I have the amino acid acceptor end. It is accepting the amino acid acceptor end. Fine. Now going ahead, which binds to specific amino acids. tRNA is specific to each amino acid. So you should be knowing this point. Each tRNA is specific to each amino acid. For example, suppose here it is U. It is UAC. So UAC, basically this particular thing will be carrying an amino acid which will be always methionine. Remember, you studied genetic code, and now one more thing. Genetic code you studied, okay? And you know that AUG stands for methionine. But over here I am saying UAC stands for methionine. So where does the AUG come from? Basically, we read the mRNA. The codons that are given on the mRNA, that is the gene code that we studied. So suppose here it is AUG on the mRNA, then the one that binds over it will be having UAC, and also it will be having methionine. So don't get confused that UAC stands for methionine. No, children, it's AUG only. We give in reference to the mRNA. Okay, so suppose every tRNA which will be there, okay, will be having a complementary codon over here in the mRNA, and they will be specific for one amino acid. It will be always that amino acid that will be there with it. Okay, so tRNA has specific amino acid. Now for initiation, there is another specific tRNA that is referred to as initiated tRNA. So to initiate something, you need initiated tRNA. What is very specific about initiated tRNA? Children, it will be always UAC over here, and it will always carry methionine over here. Okay, so that means if you remember, we have studied this thing, always the mRNA, okay, the mature mRNA will be having a start codon as AUG. Okay, the start codon will be AUG. Okay, now the stop codon can be UGA, UAG, UAA, everything. Okay, that is something different, but the start codon is one thing, that is AUG, and AUG basically will be having an opposite tRNA over here with complementary thing, that is UAC. And this particular thing always has an association with what, children? With methionine, that is the amino acid. So I am saying there is a different type of tRNA, and you call this tRNA different because it is permanent always with the amino acid and everything that it carries. It is known as initiated tRNA. So initiated tRNA has to come and attach, then only the process of initiation will be, the process of translation will be starting off. Then the next amino acid, the next tRNA will be coming with the another amino acid, and the same thing goes on. But the initial one that has to come and attach is known as initiated tRNA, and it always has methionine, children, and over here in the anticodon end, it has UAC. Now, how is mRNA read from which frame to which frame? Basically from 5' direction to 3' direction. 5' direction to 3' direction of mRNA. I am giving reference to mRNA, children. Please remember, I am giving reference to mRNA. When you are talking about tRNA, what is the side? this is, if this is 5 dash end, if you go like this, 5, 5, 5, 5, 5, 5, 5, 5. Basically from here to here it is red. So in reference to this it is 3 to 5, but on the reference to our mRNA it is always 5 to 3. 5 to 3, 5 to 3, 5 to 3. Okay. So 5 to 3, 5 to 3, it moves on, moves on. Okay, there's a picture like this. Over here this is 5 and 3. This moves, moves, moves. So everything is being read in this particular direction itself. Okay. Now, okay. Now, there are no tRNAs for stop codon. So, I cannot expect a tRNA there for a stop codon. So, stop codon is not being having any tRNA. That means there won't be any amino acid also, which is why in our genetic code thing, we have always studied that this is UAA, UAG and UGA means stop. It is not responsible for any amino acid because there is no tRNA. There is no tRNA which will be specifically responsible for amino acid for this particular codon. Okay, please understand that. So, which is why decision there are no tRNAs for stop codon. Nothing. On stop codon, you won't be finding a tRNA. Okay. Now, the secondary structure of tRNA has been depicted that look like a clover leaf. Okay, that looked like a clover leaf. This was depicted. Okay, but then an actual structure, the tRNA is compact molecule which looks like inverted L. So, you see, you just think that this is the L that we have, but this is kind of inverted thin structure that we have. It looks like this. Okay, this is an inverted L structure that you see over there. Not exactly like this, but yeah. Oh, you. Okay. I meant like this. Okay. Now, going ahead. So, we know it's clover leaf shaped if we say the secondary structure is clover leaf, but actual sense, in actual sense, what does it look like? It looks like an inverted L. It's also a very important question. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA. What do I mean by this? Basically, if I say that there is a tRNA, if I say that there is a tRNA, basically children, it has to attach to an amino acid. It has to attach to an amino acid, and this particular process requires the presence of ATP. It is a very high energy yielding process. Okay, and this process is known as charging of tRNA. Okay, this process is known as charging of tRNA, and also it is known as amino acylation of tRNA. Amino acylation of tRNA, as in when this tRNA is getting one amino acid, you call this amino acylation of tRNA, or you call it charging of tRNA. And please, even in your dead sleep also, you should be knowing that this is the first step of translation. This is the first phase of translation. Just think of the situation where the tRNA is coming but without an amino acid. Is there of any use? No, it's not of any use, because these amino acids are the ones that will be coming together to form what? A polypeptide. A polypeptide, or later a protein probably. But they have to come together, and if the tRNA is not carrying the amino acid, what's the use of it? So the first phase will be having charging of tRNA or amino acylation of tRNA. This was not given in the para, I've just changed because anyway you're talking about tRNA. So, you know certain things about tRNA as of now. That is, it has an anticodon loop with an anticodon at the end. Okay. It also has an amino acid acceptor end, where it is waiting and accepting the amino acid. Now, this process where the amino acid is going and getting onto the tRNA, is getting bound to the tRNA, you call this as charging of tRNA, or you call it as amino acylation of tRNA, and this requires high amount of energy. So, it occurs in the presence of ATP, dear children, and this is the first phase. It is occurring in the first phase. Okay, this is occurring in the first phase of translation. This is a very, very important question. I will show you the question as well. Now, the first tRNA that is there to come and attach to the mRNA, you call this tRNA as initiator tRNA. It will have this code in the anticodon, that is UAC. It will have UAC in the anticodon I'm telling, okay, that means in the tRNA, and it will be having methionine. Okay, so always all polypeptides will be having methionine as the first amino acid. Ma'am, what about the stop codons? Stop codon is not read by the the tRNA. Okay, the stop codon is not read by the tRNA, and if we talk about the structure, it was initially said that secondary structure. Okay, so in the case of everything has primary structure, secondary structure and tertiary structure. That means more complex it gets. Okay, so according to this, the secondary structure of tRNA has been known to be clover leaf shaped. Okay, clover as in you might have it at the end of your food and all. No, sometimes you just Google it once. Okay, you'll get to know. So clover leaf shaped, it was initially said like that, but then later on it was said it has a very compact structure and it looks like inverted L. Cool. Does it work? So this is all about your tRNA. Please remember the first phase. Again, I'm telling you, it is amino acylation or the charging of tRNA. Cool. Now let's go to the structure of the ribosome. So ribosome consists of structural RNA and this and that and things like that. So ribosome, as I said, it looks like a sandwich. So over here we have a larger subunit. Okay. And then over here we have a smaller subunit. Okay, and I am saying that this subunits basically are made up of two things. First thing, proteins, and then I say structural RNAs. Structural RNA. So how many different types of proteins are there, children? It's a very important question, came previously. So it is 80 different types of proteins which are there for the formation of ribosomes. Now structural RNA, you have rRNA, where R stands for ribosomal RNA. Where R stands for what, children? Ribosomal RNA. Fine. So over here you will be finding RNA, over here you will be finding proteins as well. All of them together form the ribosome. The ribosomes, if they want to be in this state, that is bound state, so you need the presence of certain ions which you call as, there are certain ions over here, and these ions, what do you call, children? You call these ions as magnesium ions. Magnesium ions, that is Mg2+. So Mg2+, it should be there. That's when they can be remain in the bound state. And this is this has been asked in the previous year question paper as well. So you have to be very careful with this. I know it's not given in your NCERT, but as I said, if everything is given in your NCERT, then everyone will be scoring the same. So we do not, obviously they do not want that. Okay, so which is why certain things will be outside the box and you have to be very much prepared for it as well. Okay, cool. Now, in the inactive state, it exists as two subunits, that is large and small. When the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. So what happens? It's not that both of them, they come together and then they see the mRNA. No, it's not like that. So the first step of the process of translation, please do not get confused with the first phase of translation. This is the first step. First phase, as in this was happening. In the process to begin up, you need certain things to happen, and that is what you mean by charging of tRNA or amino acylation of tRNA. That is happening in the first phase. Well, this is the first step. Now everything is set. Now we just have to start off the translation. And when you say you are starting off the translation, that means there is a small subunit, and this small subunit has come in contact with the mRNA. Has come in contact with the mRNA. So this is the first step. Now if you are asked about the second step, what is the second step? A tRNA carrying the amino acid, that is methionine, which is the initiator codon. So this is basically the initiator tRNA that comes and binds. This will be the second step. And later comes our ribosome's larger subunit. This is the third step, children. So first step is when you have the small subunit comes in contact with the mRNA. Later on, the second step will be what? Basically the tRNA coming and binding on this, and then comes what? The larger subunit. Please know this particular thing. Okay. Now over here there are two sites in the large subunit for subsequent amino acid to bind to and thus be close enough to each other for the formation of peptide bond. What is happening, ma'am? So basically, if I say everything is set now, okay, this is a small subunit, this is the large subunit that I have over here. Okay, just a bit of corrections. This is the small one, right? Now over here I have the mRNA, and this mRNA has a UG. This is 5' end, this is the 3' end. After that it has GGU. Okay, it has GGU. Now let me just see what does GGU stand for. GGU will be coming over here. GGU is for glycine. Okay, cool. Now let's see. So the first step will be the initiator codon has to come and attach. So initiator codon comes and attaches, but this is in one particular site, okay, and this site is known as P site of tRNA, of what? Ribosome. Similarly, there is another site, you call that E site of ribosome. Then there is one more site which is known as A site of ribosome. So initiator codon, it always, initiator tRNA, please understand I am talking about initiator tRNA, okay. It comes and attaches over here in the promoter thing, okay, in the P region, not the promoter thing, and over here what we will be having, UAC. And this will always have one amino acid which is known as methionine. Now the next tRNA again will be coming in, next tRNA will be coming in, and this tRNA will be having an anticodon for the next site. If GGU is there, then it will be CCA, and GGU basically stands for glycine, okay. So in similar way, different amino acids will be coming and joining, but different tRNAs will be coming and joining, but we want what? We want amino acids to be coming together. Over here we see there is amino acid here, there is an amino acid here, but they have to come together. How does that happen? So for that there will be enzymes, children. There will be enzymes, children, which will help in the formation of a bond over here between the amino acids, and this particular bond, what do you call this bond as? This is known as a peptide bond. While doing this, they also make sure that slowly this bond is lost. The whole tRNA got lost? No. So slowly what happens, basically there will be a bond over here with the tRNA, right? This is entirely lost. Now this M is basically bound to the glycine. Now what will happen, children? You have to use your imagination power. This particular ribosome is now going to shift this side. This particular ribosome is going to shift this side. So what will happen? This particular tRNA will be coming to the promoter side, and there will be space for another tRNA to come over here. Again I will repeat what will happen. Basically this particular ribosomal units will be shifting towards that side. When they shift towards that side, what will happen? This tRNA will move here, this tRNA will move here, right? This tRNA will move this side, this tRNA will move this side. Now what do we have as a vacant seat? That is A. Now to A, another amino acid will come. To A, another amino acid will come. So now I can draw something like this. Over here you have the large subunit, you have the small subunit, okay, and you have this thing where AUG was there, and then GGU was there. Now suppose it's CCA, okay, and there are three regions. You have E, you have P, and you have A. This is A, this is P, this is E. So to E side what came, children? To E side that tRNA which was having the methionine came. So this E is over here, the tRNA which was having methionine is over here, but as of now it does not have methionine. The methionine is gone, it is given to the next codon, okay. So over here what has happened, this E is there, E means exit. So from here the tRNA will exit, this tRNA will exit, and now will be binding to another methionine which will be there, okay. And the same thing happens, again it will get charged, again it will come again to form a bond whenever necessary, okay. Now going ahead over here, when I shifted this, I also made sure I shift this to this. So here I have the codons, anticodons, and I also have glycine. But this time glycine is with what, children? It is with methionine, which I got from here. Again another tRNA will come over here. Another tRNA will come over here, bind to these codons, will have some another amino acid. These and this, they will form a bond, and here, the bond which is over here, it will now break. This particular bond breaks, and now these three guys will be together. There will be peptide bonds formed between these three guys. So what you are getting is a chain of amino acids. A polypeptide is being formed. Over here, what are they forming, children? They are forming the peptide bond, and whenever they are forming one bond, they are breaking the bonds over here, so that the amino acid chain is not connected. And you can see the same thing over here. So to A side something comes and attaches, the bond transfers over here, and now you have a chain of amino acids. You get it? So A site is free, every time it comes makes a new thing. Cool. These are the steps, and this is very much important. So for initiation, the ribosome binds to the mRNA at the start codon that is recognized only by the initiator tRNA. So initiator tRNA comes and attaches. Now the ribosome proceeds to the elongation phase of protein synthesis. Now it is going to make the protein, so it is known as elongation phase. Okay. During this stage, the complex is composed of an amino acid linked to tRNA, sequentially bind to appropriate codon in the mRNA by forming complementary base pair with the tRNA anticodon. I said this something only. Okay, they are forming base pairs and they are coming together with, and this particular tRNA is attached to an amino acid. Cool, that is what they have written in a complicated way. The ribosomes move from codon to codon along the mRNA. The ribosomes, they are basically, as you can see, over here they are moving. Okay, over here they are moving, sliding towards that side. So amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. So this thing is actually mRNA, but who gave this information to form this? Obviously the DNA. So DNA transcribed into mRNA, and then the mRNA is now helping in the formation of protein. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosomes. At the end, what is happening, children? A release factor binds to the stop codon. So at the end, just imagine that there is a stop codon. Stop codon as in whenever you have UAA, UAG, UGA everywhere. Okay. So at this point of time, tRNA does not comes and attaches. You have already studied about that. Then what will come and attach, children? There will be something else which is known as, children, release factor. It is known as release factor, which says that, hey, whatever is done, whatever amino acids are being made, just release it. Just release it, and now you have a polypeptide chain. Okay. So on the stop codon, the tRNA does not comes and attaches. It is the release factor that comes and attaches. So I am going to underline it over here. Okay. Now, a translation unit in mRNA is the sequence of RNA that is flanked by the start codon. So, what do you say as translation unit? How do you understand this is the area where translation has to happen? So, obviously mRNA is there for translation, but mRNA is pretty long, right? So, mRNA will have translation wherever they see that there is an initiator codon and there is a terminator codon over there. So, this particular unit that you see over here, this will be the translational unit. Translational unit, children. So it is flanked by one start codon and there is another stop codon over here on either sides. Got it? Now, an mRNA also has some additional sequences that are not translated and are referred to as untranslated regions. So can it have introns and exons? No. Okay, exon it is there, introns is being removed. Introns is something that is comes in between, does not code for anything, so it is being removed when mRNA is being formed, right? So this particular mature mRNA does not have anything like that, but there are areas which remains to be untranslated. So this untranslated areas are either here or here. Either here or here. What do you call them? Untranslated regions, or UTR. Okay. You call them UTR, or you also call them untranslated regions, children. You also call them untranslated regions. Now the UTR are present at both 5' end and 3' end, that is before the stop codon and before the start codon and after the stop codon. Okay, they are required for efficient translation process. So if they are not there, obviously you won't understand that, okay, this is the place where translation has to begin. Okay, I hope this helps. So we studied about the structure of tRNA, we studied about the structure of ribosomes, and then we saw the process of translation as well. Remember, EPA, in your book just two sites are given, but understand there is one more site, that is E, exit site, where the tRNA exits. Now the tRNA is like, I do not have an amino acid, it roams around, sees that, oh, I got an amino acid. So for example, it's AUG, it will get some amino acid, and this amino acid will go bind, this particular tRNA will go bind to a region which has the exact opposite of this, right? So it will be UAC over there. Okay, cool, works, children. Now let's start the questions over here. Try to answer and do let me know in the comment section. The first phase of translation, I'm not asking about the step. The first phase of translation is binding of mRNA to ribosomes, recognition of DNA molecule, amino acylation of tRNA, recognition of anticodon. This came in NEET 2020. Do let me know the answers. I am going to solve all the questions towards the end of the video. Okay? Going ahead. Many ribosomes may associate with a single mRNA to form multiple copies of polypeptide. So it's saying that there could be one mRNA, and this mRNA is basically bound by multiple ribosomes. Okay, many ribosomes may associate with single mRNA to form multiple copies of polypeptide. Why it is happening? Because I want to make, okay, so basically what is this ribosome doing? Making a protein. Here also, this is also making a protein, this is also making a protein. So this is read 3 times because it is that much amount of this particular protein is required. So this is a protein being made, and this protein is in higher requirement. So which is why there are multiple ribosomes doing the work. Okay, so such strings of ribosomes are termed as, even though I didn't teach you as of now, but I wanted to teach you, which is why I put this question. So you can try for yourself also if you are aware of this thing. Okay, you can give a guess. Let me see who all knows it already. Okay, so do let me know the answer. Now going ahead, the 2 subunits of ribosome remain united at a critical ion level of. Who is that ion that plays a major role in the bounding of what? The 2 subunits that we have. Who is that thing? Tell me, tell me, tell me. Now, fourth, amino acid sequence in protein synthesis is decided by the sequence of. So the sequence of amino acid, that this amino acid has to be made, then this, then this. Who gives this sequence? Who gives this particular sequence? Who is responsible for this? Do let me know. Okay. Now last question. The RNA that pick up specific amino acid from amino acid pool in the cytoplasm. So amino acids are like flowing around in the cytoplasm. From there, someone picks this amino acid. Okay, and let's go to the ribosome during the protein synthesis. Who is this? RNA. Which is that RNA that is basically carrying this amino acid from the amino acid pool? It's a very easy question, no? Came in 1987. Okay, these are the recent questions that you find over here. Okay. And the question has also come about the proteins, the number of proteins. That is why I told you to please remember it. It's 80 proteins. Okay. Now let's try to solve the first question. The first phase of translation is, children, amino acylation of tRNA. I told you first thing that we need is the tRNA, if it's there, it needs to have an amino acid group attached to it, and this process is known as amino acylation, and this is occurring in the first phase of translation. Okay. Second thing, many ribosomes may associate with single mRNA to produce multiple copies of polypeptide simultaneously. Such strings of ribosomes are termed as. So basically what is happening, in one particular mRNA there will be so many ribosomes coming together because we need the formation of so many proteins as well. Well, this structure that you see, it is known as polysome. What do you call it? Polysome. There are different type of questions which are being formed around it. So you have to be preparing yourself really well with this thing. I will be asking in mock test in a different way. Okay. Now, the 2 subunits of ribosome remain united at a critical ion level of magnesium. I already told you magnesium is required for the larger subunit to remain really close with the smaller subunit. Okay. Now, amino acid sequence and protein synthesis is decided by the sequence of rRNA, tRNA, mRNA or cDNA. It's the mRNA. If mRNA says AUG is first and CCCA is first, then according to it only the amino acid which will be coding will be formed. If there was a change in the mRNA, obviously this will also change simultaneously, right? Now, so the answer is mRNA. The RNA that pick up specific amino acid from the amino acid pool in the cytoplasm to the ribosome during protein synthesis. So there is only one RNA which does this, okay, which has very close association with amino acid, and that is our tRNA. So you know tRNA has an end which is known as amino acid acceptor end, where the amino acid comes and binds, and this requires lots and lots of energy, and there is only one person to spend this much amount of energy, that is tRNA. Okay, works. Now one question that I want to ask you as a homework I am giving you, that is this thing. In protein synthesis, the polymerization of amino acids involves 3 steps. Which one of the following is not involved in the polymerization of protein? First is termination, second is initiation, third is elongation, and the D one is transcription. Okay, the question is which one of the following is not involved in the polymerization. Polymerization basically means in the formation. Okay, when we say that this and this come together to form a polymer. To form what? A polymer. Okay, that means that this is a monomer. This is a monomer that is going to form this polymer. Didn't understand? So basically this is a monomer, this is again a monomer, this is again a monomer. So many monomers. So many monomers are coming and helping in the formation of a polymer, and this whole process is known as, children, polymerization. Formation, as in formation. Okay, I hope it's clear. So that's all. Do this question and do let me know. Okay, and if you think that this particular video was really helpful for you, then do not forget to subscribe to our channel, and I wish you all the very best. Be happy and give your maximum while you're studying. Okay, give your best and everything is going to be really, really good. So bye bye dear children, bye bye, take care.